and I'll hand over to Daniel.
Hi, hello. So my name's Daniel Iddon. I started Woodland Grow off the back of a tree production innovation fund that was funded by the Forestry Commission over the last 3.5 years. So Woodland Grow is the spin-off, I suppose, from about 5 years worth of work where we have tried to bring and mimic woodland soils into tree production and afforestation. And it really started for me 5 years ago when I bought a woodland and I grew my own trees in my own nursery, with third generation horticultural background. And when I planted all the trees in the April, they all nearly died. So I've had nice healthy trees in the nursery, plant them in the woodland, and they all nearly died. And there's a nature gap that we have, where we're growing trees with synthetic fertilisers, the nice NPK slow release fertilisers that are plant available, but they're synthetic. And then we've got the woodland environment, which is fungi, bacteria, nutrient cycling microbes. So we've got a nature gap. So the trees recovered, they grew, and that's what I'm sure you're all familiar with, and that's the ecological process whereby trees give up their sugars via the exudates to the roots, and then they attract the fungi and the bacteria, and they start doing a nutrient exchange. So everybody's familiar with that process, not least the Forestry Commission, and this project, which we're here to talk to you about today as an output, is how do you bridge that gap? How do you get a tree to grow as quickly as possible from the nursery into the land that you're planting in, which tends to not have much fungi in it. So throughout this presentation, we're going to be talking about what a tree needs and how does it get it and where does it get it from and how we've gone about doing that. So the project was called Living Soil, Seed to Woodland, and this is how we're bringing it through. So the basic premise is, how do you make best use of residues within forestry? So after you've managed a woodland, you end up with some timber, and after you've taken the timber away, as an industry, we've worked out what to do with the particles. So we've got panel boards, we've got structured boards, we've got bark, we've got chip, and bringing all those things together, you end up with bark fines that are left, and typically that would get used as a peat-free grown media mix. But what we wanted to do was take those residues from the woodland, repurpose it, and bring it back into growing the next generation of trees without using peat or synthetic fertiliser. And to do that, we have to start thinking about the microbial community that the trees need. So people will call them weeds, but I think if you look at them in a biological sense, they'd be plant indicators of the soil health and the condition. So if you've got weeds, it's because you've got a bacteria-dominated soil. And as you walk through the succession, you end up going through your agricultural crops, through to your maize, and it's at that point that you have an equal ratio between your bacteria and your fungi. So that ratio is 1:1. Maybe when you're growing lettuce, it's 0.75 to 1. When you're then looking at your ericaceous plants and your rhododendrons, your azaleas, you're probably at about 2:1. Your fruit and nut, it's about 5:1. Broadleaf, it's 10:1, and conifer, it's 20:1. So if you're planting trees in soil that's previously had grass on it, you don't have enough fungi in the soil for a tree to start growing. So that's the fundamental problem that we have and why we have this nature gap. So healthy tree in a nursery with synthetic fertiliser, and then soils that perhaps don't have enough fungi and are the wrong type of fungi for the tree to start doing a nutrient exchange. So it has to give up sugars, as I said, to attract the microbes to the feet of the tree. And that's where you normally have this 2-year gap before a tree starts growing. And then during that period, I think you're all familiar with a tree under stress, whether that's been pest, disease, being eaten by your rabbits or your deer. And that's also because they've been grown with synthetic fertiliser and good N. A lot of N makes them nice and green and big, but also makes them quite sweet and tasty. So the bit that we look at is what we call the poop loop, which is if you've got the organic matter, most soils have got plenty enough bacteria. And then as you build up your microbial community, you've got the protozoas and the nematodes, and then you've got the arthropods, and then the birds, and then the animals. But it's through here that you need the fungi and the bacteria, protozoa and nematodes all working together as a full cycle. So there's plenty of enough nutrition in almost all soils around the world, but there's a difference between the microaggregates and what is plant available. So to be plant available, it needs to have been eaten by the bacteria. Bacteria needs to be eaten by the protozoa. Nematodes eaten the protozoa, and then you end up with what is effectively these enzymes, these extracts, and then the fungi and bacteria do a nutrient exchange with the plant. So the plant gives up some sugars to attract the fungi bacteria, and they do an exchange. So if a plant's got a choice between synthetic fertiliser, which is free food, or food that it has to give food up for, it's always going to take the free food. So that's why nurseries tend to use the synthetic fertiliser, because it is a consistency with the growing and getting a crop. And that's why we have a gap in the natural world, because that's not how nature works. So how do we do that? How do we bring that into what we're doing? So we effectively breed a microbiome. And within this picture, this is what we see under the microscope. Is the lungworm is a nematode. We've got the long hyphae, which is the fungi. We've got bacteria, organic matter, protozoas, and nematodes, the other microbes, sorry. And then you've got amoebas. And all of this soil food web is what happens naturally in the world. And what we've effectively worked out how to do is breed a community of microbes and then add natural sources of nutrition that both feeds the microbes as well as feeds the plants. So those ingredients are your typical NPK, which are your macronutrients. Then you've got your micronutrients, which is the likes of the sulfurs. And then you've got your trace elements, which are the likes of borons and molybdenums and zincs. So the plant needs all of those things. It's not just NPK, it's your macro, micro, and trace elements, but so do the microbes. So by layering natural sources of nutrition, minerals, and trace elements, you end up being able to feed both the plant and the microbes. And to do that, we take our recipe, which is typically parts of a tree, and then things like hemps, silage, straws. We put them in a bag and we time mature them in a woodland, and that stays there for typically 12 months. During that time period, we're monitoring that for microbial communities, the fungi, bacteria via the microscope. We're doing nutrition analysis, looking at what is the food and the communities that are available for that. And then we're also doing the DNA analysis to work out what is the communities within those groups. So it's relatively complex in terms of, it's not just about fungi and bacteria, it's the groups within those. So what are the types of bacteria? What are the types of fungi? What are the ratios between those individual groups? And then how do they work together? And whether it's a diverse community, a complex community, or a simple community, makes a difference as to whether you're going to be growing a weed or a tree. I can see some questions coming through, which is good. I'll probably try and get to the questions at the end and come back through to my slides. So as part of this project, what we've been working on is how do you make this growing medium? How do you create this community? How do you bring in your nutrition? And then what we've also been doing is growing with those plants. So we've grown in my tree nursery in Wales, which is located in my woodland. We've been growing with Forestry England at Delamere, and we've been growing with Alba trees in Scotland. So we're looking at geographical differences, we were looking at climate differences, we were looking at different growing systems, whether it's the elepots, which you can see in this picture, which is a biodegradable paper pot, which is breathable, versus normal root trainers that's typically used, and then whether it's in a polytunnel outside or in a greenhouse. And what we found was that the trees were healthier when they are grown with a diverse, complex community of fungi and bacteria. In this picture, we've got an oak on the left and some Sitka on the right. And just to give you a broad range of kind of what we're doing, so we've got some hazels in this picture. This is the ella pot, and then a hazel that hadn't germinated, but within that, this is how we see the fungi. You don't often see what's going on. If you see a fungi sprouting on the top, that's one, but there's probably 1,000 that are underneath. And that's the other thing about fungi and microbes, is it's not the fruiting fungi that's the one doing the job per se, it's the one that's actually become dominant to get to the surface, and that was the one that will stay there. But the example I had in the picture earlier on was one fungi came to the top and was fruiting, it filled the bag, but there was actually 1,000 underneath. So complex communities are more than what you can see. So one of the things that we've seen with our growing media and our growing trials is that the oaks were mildew free. Mildew being an airborne pathogen, we have it at the nursery from previous growing of our oaks before we started this project. And in our controlled growing trials, we had trees growing with our living soil, and then trees growing with what is a typical product, and in this case it was Jiffy. It was a tree-specific grow medium. And on the left-hand side, as I see it, we've got mildew-covered oaks, and on the right we've got oaks that were in the same polytunnel for the same period of time that are all clean from oaks. So this isn't me saying that the trees can resist pests and diseases, but this is saying that if you've got a diverse microbiome and you've got a diverse source of nutrition, and there's elements in there that they don't typically get access to in the synthetic fertiliser NPK version, you end up with a change in the leaf form, its biological resistance, and the physical construct of the leaf changes. So this is an example of that. When we grew the hazels, we found that the hazels that were grown in synthetic fertiliser, again with Jiffy peat-free, was covered in aphids, and then the oaks, hazels that were grown in our living soil were aphid-free. So there is a difference between how you grow them and the state that the trees are in. So that's being under pressure, being under stress, needing more water, all of those things come into play. So using our compost, I think we had every 4 days there was 5% more water retention. So if you accumulate that over a month, we had 25% more water retention over a month in our nursery using our living soil than we were using the Jiffy equivalent. So one of the things that we wanted to try and do to understand how we were doing it was to use lettuce. Might seem a bit of a non-specific plant to try and do it, but lettuce are very nitrate hungry. Within the 20 days that we grew these lettuce from seed to the picture you see here, that's the same amount of nutrition as a tree uses over 12 months. So when we're growing our trees, it's all the nutrition, all the biology is in with the roots, and the tree grows. There's no additional foliar feeding, and we're using less water, no more food. And it's the same when we did lettuce. We were able to grow the lettuce for the same duration, same period compared to peat-free, but they had to use additional foliar feeding and additional water. And it's the same recipe. It's also the same recipe when we did primroses and we did some wildflowers, scabious devil's bit. And in this picture, the top was the Woodland Grow compost, and the bottom was proprietary media from Melcourt. And within that, you can see that there was more green in the leaves, there was better growth, and there was better root structure for those. And again, it was the same recipe. And I think the thing here is key, is its fungi is doing the nutrient exchanging. So you can't overfeed a plant that's got fungi doing the nutrient exchange. Whereas if you're giving synthetic fertiliser to a plant, it's always going to take what you give, or it leaches and it ends up down the river. So that's the other point within what we're doing, is we're trying to be as sustainable and circular as possible. So all of the materials that we're using are residues, and they're all procured and sourced from the UK. As I said, the bark fines, the residues from agriculture and fisheries that give our nutrition are all sourced, and it's made in the UK for the UK, and specifically for trees with the addition of fungi. We've also grown agricultural crops, so we've been working with the likes of Andy Cato at Wild Farmed, and we've done some spring wheat. In this example here, we've been working with some winter barley in Cumbria. Similar kind of message, really, that for this application we applied our nutrition biology in April, and then we didn't apply anything else. And then farmers typically applying fungicides and hormones and growth regulators, to Epsom salts, trying to keep and manage the crop. Whereas for us, we applied it once and nature did the job. And it, again, it's the same recipe, but with just additional points of N. So how do we do this? So when we've been doing the DNA analysis, what we're finding is that if this is a Shannon and richness graph, and we've got our recipes, recipe 1, recipe 2, recipe 3. We've got composted bark fines on its own, and then we've got Jiffy that we used as part of our growing trial. And to a statistical significant difference, we've created a richer, more diverse community. So not only have we got more fungi and lower amounts of bacteria, we're able to look at the community and it is more diverse, which means you get more function as well. So I think a lot of people would think about the community, the size, the complexity, and I think within this 16S is the bacteria, ITS is the fungi. You can see that what we're working with is 40% unknown fungi, and that's because the scientific community has only identified 110,000 species of fungi in the world, and it's estimated by Kew Gardens to be 2.5 million species out there. So we're only working with 6% of the population, and therefore if you're going out and collecting a particular strain of fungi, as an example, you're getting a very, very, very small percentage. So I think the way to think about this is, if you give the tree enough diversity, enough abundance and enough function, it's able to actually talk to and work out which microbes do the job for it, which is what we found when we've been growing our oaks. And we had various recipes from various sources because we were trying to work out whether, for example, a hemp-based microbiome versus a silage-based microbiome would create a community that was different for conifers versus broadleaves and oaks versus cherries. As I said, the project was over a few years, and it's quite a sizable project. But what we found is that the core microbiome, when you actually boil it down to what is the tree talking to, common through all of these, it was 4 fungi and 30 bacteria that was the same, and it didn't matter which recipe that it came from, or whether it was bark fines on its own. So I think there's a point here where if you give enough abundance and enough choice, the trees are able to choose which is the microbiome that it wants to negotiate with and have that symbiotic relationship with. What is giving up its exudates to attract those fungi and bacteria, and therefore, for us, we expect a difference between oaks versus aspen, for example. And I know for anyone who understands trees, there is a lot of companion growing, so whether that's oaks and cherries or Douglas and birch, there is always a community or group of trees that grow well together. And for us, we think that's more about the microbial community and how that is working. But again, this is a relatively expanding and exciting part of fungi, mycology, nature, and trying to work with nature, not against nature. So if I was to, this is my last slide, I was trying to keep to half an hour so we can have some questions and answers without taking all of your lunch. So on the left-hand side is a picture of the pellets that we've created, and effectively that is our compost that we've reduced the moisture and then we've pelleted. So we're keeping the microbes alive with the moisture, and then they can be spread onto into the hole when you're planting, or onto the top of the tree area if you've planted it last year or the year before, or it's a struggling specimen tree. So the idea with the pellets, as an example, is that you can inoculate your bark, you can inoculate your mulch, you can inoculate your topsoil. It's there to breed and provide function, nutrition, and organic matter for any of your tree planting projects, your landscaping projects, anywhere where you've got plants that you're trying to transition from a nursery into their location. Highway planting, where they tend to work with a lot of bare earth. You know, there's no surprise that we have failures on the A14 because there's no fungi and bacteria, and when you plant the tree, they're just left to survive for themselves. I think it's important to note that the materials we're using are virgin materials. We do work in accordance with the regulations, even though they're not applicable, because we think that's the right thing to do. The material that we source is Grown in Britain certified. And from either a nursery or a growing perspective, or from the way in which we're working, is also plant healthy, in accordance with plant healthy. So whether it's compost or pellets, it is essentially the same material but in different forms. And just as an example for the DNA work, there's an example here from Jiffy. And we've got composted bark fines on its own, and then we've got compost recipe 1, 2, and 3. And the reason why I use this slide is just to show that this blue is an unknown fungi. So that's fine, it's between 20 and 40%, depending on the replicate. But there is one tree here which is growing, which is our oak, and it's 100% unknown. So for us, we don't believe in taking one species that we know about and applying it and using it, because it's like a recipe and a cake. It's complex, there's lots of different ingredients, there's lots of different functions. So whether it's particle size or the proteins, or, you know, it's like you can't take an egg, put it somewhere else, and then try and make a cake. You've just got an egg. So the thing for us is about that community, abundance, diversity, and using materials in a natural way to bring what a plant needs. And I think before I try and wrap up and go to the questions, is to just reflect on, it's the same recipe that we've been using throughout our growing trials, and it didn't matter which plant it was. The thing that was consistent was it was fungi. So whether it's an oak, whether it's a hazel, whether it's a lettuce, whether it's wildflowers, whether it's ornamentals, crops, there's an ecology here that's working. And, you know, fungi are older than oil and gas. You know, fungi came out of the water, they came onto land, made a relationship with algae or bacteria to create lichen. Lichen break down the rock, rock created soil. So that's why we have, you know, the relationship. It's not just a symbiotic relationship. It is how trees, it's how plants grow. So if you're using and planting this winter, I hope that you will consider what we've done here and think about what a plant needs in terms of, does it need fungi, does it need bacteria, not just is it a synthetic form of NPK that's available in a dry powder, because ultimately that's not what a plant needs, because if it does that, it's delaying the fact that it needs to start talking to those microbes, because that's how it will ever move forward. So if you're trying to get a tree to grow healthily and happily, not stressed, consider how you create that fungal population and what source did it come from. So if you've got a compost made out of food waste, it will be a different microbiome and what it will grow to if you had a a compost and a microbiome that came from a woodland. And I think that is the thing to think about with that succession line, if I go back to it, is the microbial community, the complexity of the food, where it comes from, all makes a difference as to how the trees grow. So for anyone who's on the call and wants more information, I'm happy to have another session, either one-on-one or groups for your communities or your organisation. For anyone who's interested in coming to have a look at our woodland and our nursery where we make all of this, that's in Pembrokeshire, happy to arrange that. And I think for any questions you have, we'll try and cover them in this session. Or if you ask Green Tech, then they can come through to us and we'll either get in contact directly or we'll provide the answers back through to Green Tech. So this is new. It's not come from a project where we're trying to sell poultry manure because we've got a big poultry farm. This is very much about how do you grow a tree and reduce that gap between nursery and afforestation and planting, where a tree doesn't have any fungi but needs fungi. And it is as profound and as simple as that. So I don't know if that was a good, useful half an hour. I hope it was. It's been 5 years of our lives, and a whole team of people that have brought this. I'm certainly grateful, and I am, for you listening for this last half an hour.
That's great. Thank you, Daniel. That's, yeah, really good. We've just got a few questions here, so we'd like to cover those off. So the first one, which I think would be one for you, Daniel, would the genetics be location specific? Is there a potential issue for limited genetic diversity?
It's a really good question, and I'd probably like to explore that a little bit more. But if I talk holistically, very briefly, that it starts off with a seed. So where that seed is collected, and it's like the human microbiome, the gut microbiome, the mum, the relationship with the baby is no different to a seed from that woodland. So that seed is covered in microbes, and that community is specific to that woodland. So that's important, and maintaining that. And we can, and this isn't something that we've offered here. You know, we've done a catch-all, one-size-fits-all product here, but we can effectively breed that microbiome from that woodland. So if somebody had a specific SSI woodland or one of concern and they wanted to extend the canopy, it is totally feasible for us to do a custom blend, work with a client and create that microbiome to go with that seed and that seed zone. But I think one of the complications from that is to make sure that that seed and the seed zone and that area of that woodland is healthy, it's functioning, it's got the nutrition it needs, it's got the diversity it needs. And I think I've seen it in another question. What you tend to find is that trees that aren't managed, and we don't really manage woodlands like we manage farmland, we don't manage the biology, we don't manage the nutrition, we plant them and walk away and look after them in that sense, but we don't intervene and try and add more carcasses to the root of the tree like we were talking with some of the Scottish research. And this is the interesting thing about our nutrition, is it has hoof and horn, it has bone meal, it has blood meal, it has all the things that you would expect. It has fish, it has the limes, the calciums, the phosphates and the potassium sources from plants, whether it be hemp seed or whether it be soybean or whether it be alfalfa. There's all these nutritions that overlap on each other. And I think if we go back a few hundred years, we'd have been skinning animals underneath a tree, and there would have been all these kind of nutrition going into a tree. So for me, seed zone and where and how you manage a woodland all come back to fungi, bacteria, and nutrition profile. So we do nutrition analysis to look at what is in the soil and what is plant available. And we've actually done some interactions with large estates recently where they're struggling with big trees. So they've got these 200, 300, 1,000-year-old trees, and they're struggling. How do we help? So within that scenario, we do microscopy of the soil, we do nutrition analysis, and then if we want to go as far as we can, when we do DNA analysis. And with those three things, you get a picture of what a tree is lacking and then you're able to put back what it needs. Now, some of that might be diversity of function because the fungal community is dominated by one class, and if that was the Leomycetes, I think that's pronouncing it right, well, that's the one full of all the molds. So maybe, by increasing diversity from other functions and other fungi groups, you're able to bring what is in balance. So we don't see good fungi, bad fungi. We don't see honey fungus being an issue. What we see is honey fungus, as an example, being dominant. And what we need to do for those cases is not get rid of the tree that's been impacted, is to actually add functionality, add diversity to the soils. And by having things like, and I know people like this as a buzzword, but like mycorrhizal fungi, they're known for doing a particular job, that's correct, but they only have an association with a living host. So one of the things that we've done with our growing trials is we noted that the microbial community increases with age as it's interacting with the plant. So this picture is a before and after. One was a compost and one is after the tree's grown for a year. So you can see that the trees interacting with the microbes over the year changes that community, and that's also true of a early, intermediate, and late succession woodland. That what a tree needs in its early days is not what a tree needs in the late days. So I think I probably rambled a little bit, but genetics, to a certain extent, is location specific, because the east of England, the west of England, the south of England, and Scotland all have different makeups in terms of clay, sand, loam, and the like. But one of the things that we're using to try and do this, because the plant is selecting what it uses, is we're using bark fines that's been collected from all over the UK already. So we're working with A.W. Jenkinson, and I think the size of their collection is at least 50% of the UK. So if we want to be specific, we can be. But what we've found is that the trees actually do the selecting and talk to the ones that they want to talk to. So if I've got a question here, so if applied as a top dressing, how long does it survive if exposed to the elements? So one of the things that we've been doing with take a step back. So we've done lots of trials with putting the material, the root of the tree, and on the surface. We've done trials where it's compost and mulch and pellets on its own. And again, that was multiple places around the country. We've done trials in the farming scenario. And when we did the top dressing, for example, the winter barley, within April to July is that window, the actual microbes and organic matter all went into the soil. So for us, the elements is about water, and it always being on the top, and then nature draws in as it's processing it. So for us, there's a benefit for putting it, if you were planting, make a hole, put the biology, organic matter, and nutrition into the hole. But it actually, for us, it's also feasible and operationally makes sense as well to put it on top, and then it will go in, because as soon as moisture and rain was to hit it, it will leach. And I think what we found with our in our woodland, because we use worms as part of our mother development, is we did some worm traps in our woodland, and we did living soil, we did hay, we did silage, and we just put some hessian sacks on each one of them. And then a couple of weeks later, we went back, and the living soil, the tree had actually come up to the surface with its roots and was going through the compost and interacting with the microbes, and there was tiny little bits of fungi just coming off the root of the tree. So for us, the tree knows where to go and get its food. I'm not going to say it smells it, but it can work out where its sources of nutrition are via the signals from the microbes. So if you've got a flat site and you can run a tractor on it or a quad, it probably makes sense to spread the fertiliser as you're going along. It's quicker and easier. But if you've got a restock on a site that's still got the stumps, it probably operationally makes sense to have some fertiliser in your backpack, and one or two of those pellets goes in with a tree. And I'll just go down the list again. Yes, sorry, I've got to tell you what I'm reading. So would the soil help restore failing mature specimens? I don't want to just be so quick to say yes. I think there's some consideration there. Sometimes it's physical, sometimes it's chemical, as in the nutrition, sometimes it's a biological. If you've got trees of concern, get in touch with Green Tech or contact us directly. We can do microscopy of the soil. We can do nutrition analysis of the soil. But what we tend to find, and what we've found when we've done it, is that there will be an element of compaction, so the physical side that, over years of running over, or whether that be events, whether that be road driving nearby, or agriculture, for example, there's compaction, so you'll see the physical bits. So that tends to then be missing is it's missing fungi. So fungi create the holes in the soil to provide that structure. So whether it's flooding or drought, fungi have a job. Whether it's nutrient runoff, nutrient retention, fungi have a job. Supporting older trees, I suspect there's an element of, if you've used glyphosate around a tree, that kills arbuscular mycorrhizal fungi, it's toxic, but it perversely, it's actually selects detrimental fungi and helps them proliferate. And then the other thing for glyphosate is it is a chelator and locks up nutrition, so it's never available ever again to any other plant. So I don't want to say it's as simple as applying this, but you're going to do absolutely no harm. There is biology in there, there's nutrition in there, and there's organic matter in there. And for the examples we've seen, we've had a look at the soil, there's no fungi, we've applied this, they put a top dressing on of some mulch, and then within a couple of weeks, I think it was, there was lots of fruit and fungi all over the place. So the only way we think we can help is by giving diversity, both in nutrition and biology and the function. So for us, I would absolutely advocate this for specimen trees that are suffering. But if there was one of concern, there is a way to let the data help guide you. So it's not just buy a product and hope and pray. The product will do the job, but if you want to be directed by data, we can absolutely collect the data and use the data to make an informed decision. So we've got another question here, which is, oh, sorry, and if I'm not answering the question, put a bit more clarity into another question, I'll come against it. So when planting medium standards, what dose rate of pellets have you found works best? So what we've done, and this might be just my way of thinking, is 1 cubic meter grew saplings, and I'll show you the picture of the saplings, and 1 cubic meter will do 10,000 saplings. So it's 100 millilitres per ellipod. So for us, what we've done is either in the compost, we've given a dosage rate, and it's on the data sheet on the website against the product of compost or pellets. We've then translated that to the weight of the compost pelleted, and then for every 20 kilo bag, we've said that's good enough for 1,000 saplings. Or if you're backfilling a specimen tree and you've got a cubic meter, then you're either applying the compost or the pellets. Again, one operationally might be easier than the other because you're on a quad or you're in a van or you're right next to where you're planting. There's a benefit and a trade off for each of those. I am of the belief that you can't overfeed because nature is doing that interaction. So the fungi is interacting with the tree and the plant, and each one is doing an exchange. So the plant is giving up some sugars and the fungi is giving up its food. And I think the scenario we've had is that, so that when we did the lettuce, they grew for 20 days and then stopped growing anymore. So there's only so much nutrition that you can put in a 100 mil. So the more nutrition you need, because the plant is bigger, the more you'll have to apply. But I don't think you can over apply because it's self-metering. So I don't really think you'd use 20 kilos on a sapling, but I don't think 1 handful versus 2 handfuls, if you're really in poor soil, is going to be the detriment. But again, if you've got any questions and it's site specific or there's a scenario that I've not covered, more than happy to field that question via Green Tech or direct. So I've got another question. They're thinking about utilising it within an urban tree planting project, and absolutely. I think there's a few points here, and I'll bring it round to. We're aware of councils that have got urban trees and they have a water problem, and they've got specialist teams driving around looking after those specimen trees and watering. Just on that simple metric, this product will make a difference, whether it be the compost or the pellets, because fungi do the job. Now, if you've got an urban planting and you've just covered it all over and there's not much soil exposed, there's nowhere for the fungi to come naturally. So whatever fungi was in there before is all that was ever going to be there. So putting fungi in is a good thing. Fungi hold on to water, they create that structure, and therefore there's less boom or bust from a water perspective. But I also think from a nutrition perspective, if you go back to the analogy of where is this carcass coming from, well, in Scotland that would happen because the deer would be on the floor, or the bear might be doing its business in the woods. More for Canada, I suppose. But we don't have that with an urban tree planting. So for me, this is almost like farming perspective, that you are applying the food for a tree to grow in a healthy way. So whilst a lot of nutrients are in the soils, if you've got an urban building project, that sounds to me like you've taken the foundations and you've moved the soil somewhere else, and then you're trying to repurpose it. And I think in those scenarios, unless it's the topsoil from the first 30 centimetres of the soil, it's not going to have the right minerals, and it's going to be quite clay heavy, and it'll need years and years and years to break down. So for me, that is where I'd use it, is either for ongoing maintenance of trees and making sure that they've got enough ability to do nutrient retention, apply the nutrients, and give them the biology and the function to be able to protect themselves. That biological response to pests and diseases, all of those things are relevant as to how healthy that microbiome is. And also for new planting, I think there's a consideration to think about what is the soil that you're using, where did it come from on the site, how much biology is in it, and if it doesn't have much biology in it, the tree has to give up its sugars via photosynthesis to attract the fungi and bacteria, and if they don't come, that's where they suffer, that's why they die. It's not necessarily because it was a hot spring. The hot spring comes into effect because there's not enough organic matter around the roots, and there's not enough fungi around the roots, and therefore, there's not enough of that transition. It's not a good story that I'm going to share with you, but we had some hazel trees that we sent to one of our suppliers, and we didn't say it had trees in it. We just sent some boxes of trees to go to a client subsequently, and they got put in the back of a warehouse for 6 weeks in April through to mid-May, or the end of May. So for 6 weeks, we had trees in a box with no light and no water as we had a hot spring. And by the time we'd worked out what had happened and we found them, they were all looking pretty ropey. So we took them out, stood them up, watered them, and 90% of them are alive now and growing healthily. So for us, we don't believe a hot spring is a reason for why trees are dying, because we just did 6 weeks with no water or light and the plants are still alive. The relevance is whether there was, and it just comes back to the same metric, how much fungi was there, how much bacteria was there, how much organic matter was there, and how much nutrition. And the plants will sort themselves out. I'm not being glib when I'm saying that, but that's we know a lot less about nature than we think. So can I give a figure for the percentage of increased survival rates? We're doing trials at the moment. We have got lots of replications in different parts of the country. We did some, as an example, so the answer to the question is I'll be able to tell you more when we do that analysis as we get to the end of the year, because they've had one growing year. But we did some work with the Tree Council where they were planting some trees over a good couple of kilometres of hedges in a farm in the Wiley Valley. We did the nutrition analysis and the microscopy, and it was pretty poor soil. It was just bacteria. There was no fungi. There was no nutrient cyclers. And within hours, we planted ours, and they're alive still. And for them, when they bought some bare root and planted them, they're dead. So we've got a position where ours are growing and the others are dead. So we are surviving better. But I think one of the things that we've got to remember is, as far as I'm aware, this is as close to nature as you can get it, and we can't account for everything. So, you know, if a deer grazed it, or something else that happened, that's still beyond our control. But we've put as much fungi and bacteria in as possible, enough nutrition in as possible, enough organic matter, and the trees are growing. I think when it comes to then applying our fertiliser to a bare root tree, and you planted it in late April, I think there's a question mark as to whether that's the right time to plant and whether our product is a miracle worker. But if you plant it at the right time, and the biology is able to interact with it before it starts warming up, if it's a bare root as an example, I think those are the things that we need to think about. I don't know if that answers your question, but I'm happy to kind of talk about it some more. So will the pellets help improve the structure of heavy clay soils? I think the short answer for me is yes, but I think there's going to be a question mark as to how you do it. So if you just have really clay soil and you just throw our stuff on and walked away, I think we're asking it to do a lot, because it's an inoculant, it's a fertiliser, and it's only so much organic matter that it comes with. So I think it would be about budget, it would be about what materials you have available locally, and whether you get some mulch and try and add to it. But within either a system, or depending on where they are, I do think it will play its part, because there is a way in which, you know, clay holds onto water, but you want to add some structure to it, and you want to add some organic matter, and you want to add some biologies. But from a soil structure perspective, clay's quite far down. So if you've done if they've done some earthworks and brought all the clay to the top, I wouldn't just put fertiliser in, but I would put some fertiliser, I'd perhaps put some mulch on the top, and I'd perhaps put some compost in as well. And I think these are the tools to be able to deal with the scenarios that, you know, you as, you know, potential clients and your customers, we've got to work together with nature and look at how we're trying to influence and what is the application rate for that particular. So just as an example, when we've been doing microscopy as part of this project with the Forestry Commission, we've gone from Devon to east of England to Wales to Cumbria to North Yorkshire to Scotland, and almost universally, except for one site in Wales that was managed by the National Trust, we haven't found a sufficient number of fungi, and is often zero. So the answer for me is always an obvious. If you use our material, you're always going to be doing better than planting and walking away, because planting and walking away is leaving the tree to survive on its own with no fungi on its roots, no fungi in the soils, and it has to give up its sugars to attract those. So it's instantly on the back foot, instantly stressed. And just to kind of give a numbers and a community piece, so you need 150 milligrams per milliliter of fungi, it's a way of measuring it from the microscopy, to do nutrient cycling. And we can track this in year 1, year 2, year 3, year 4 planting. You see the numbers increase, but I think we did one, it was year 8, and it was only 80 milligrams per milliliter. It's still not enough to do nutrient cycling after 8 years. And this tree here, that are, you know, just an example, had 500 milligrams per milliliter, which is as much as, in terms of abundance, from a 1,000-year-old oak that we did in Berkshire, and had a look at that. So microbial community, diversity are all relevant as to how you get the trees to function. So let me go and see if there's another question. So we've got a program for using industrial hemp, cover crops, and can have to capture CO2 into food, plant to prioritize crops, return them to the earth as biochar. How beneficial can these be to help that? Bit of a so there's 2 questions in there, and I think for me, biochar is one of those where I think the jury's still out. I think as a system, which I think you're talking about, I think you're trying to use materials and put them back into something that's useful and locking up the carbon. I definitely get that. How does that then work when effectively, if I called, you know, biochar an empty bathtub, there's nothing in it. So you've got to put the right community in it for it to be useful. So it might be a good shelter for bacteria, and that's a good thing. It might be good for water retention. That's certainly a good thing. But I think every time somebody has shown me biochar, I've shown them the benefits of the microbial community we've got, the organic matter we've got. And for us, I think I would far rather work with you to repurpose the materials you have to create a better use for it beyond the biochar and actually put it to use as a biofertiliser. And I think the other thing, just talking about the CO2, is because we are making this all from residues, when we do the life cycle carbon analysis, we only end up using about 100 kilos of CO2 for every tonne that we make. And compare that to synthetic fertiliser, which is an EU average of 3.2 tonnes. And if you're buying fertiliser from Azerbaijan, then it's about 10 tonnes for every tonne. So trying to use natural resources is a really good thing, both from capturing the carbon in the material, but also avoiding using fertilisers from another source is massively beneficial. I've not seen the data, and we've used biochar when we've done these kind of trials. I've not seen an increase to then justify the investment and the cost and the use. But that's not to say it doesn't work, and there's definitely benefits that it can have. It's just that we haven't seen that come. So the normal planting period we use is between November to March. Would earlier planting be an issue? No. For us, our trees can be planted all year round, and I think this fertiliser can be used all year round. I think if you were to have a really hot day for 3 days, because that's all that happens in the UK, I'd probably say don't use it on those days. But if you knew in the middle of June that there was some rain coming, I would happily go and apply the fertiliser and use the compost. I don't think temperature for me makes a difference anymore. On the basis that the example we had with the hazels being 6 weeks without any water and still growing. I think that's the end of the questions. I know I've rabbited on for quite a while, so if there's any other questions based on my answers, or I didn't answer your question the way in which you thought, then please let me know, but I'm going to quiet down for a second and have a drink.
Yeah, thanks, Daniel. Very, very interesting product. Green Tech are definitely excited about what we can do with Woodland Grow. We think there's so many uses where it can really help improve, the environment and improve schemes for our customers. So, we think this is going to be a really big product for us. Just like to thank Daniel for coming on today. I know he's a busy man, and thank everyone else for coming online as well. We are recording it, so we will put it on all the socials and things like that. But we're the sole distributor for Woodland Grow, so any questions either come through at Green Tech or through Daniel, and we'll be happy to help and help you learn more.
So we've got one last question. So you'd like to know more about using a tree nursery and a planting programme across Cardiff? Absolutely no problem. So if it was supply of Elapots, we can supply Elapots. Again, get in contact with Green Tech, just like the picture. We can fill trays up with Elapots, so you can use them in tree nurseries, whether that's a community nursery, a commercial nursery, volunteer-based nursery. Comes with these paper pots, comes with a grow medium, and it's there straight away to go use. And for any tree planting projects where you want to buy the fertiliser and then use it, if you've got questions about that, absolutely no problem. Just drop me an email. So it's daniel@woodlandgrow.co.uk. I think there's one last point that I'll try and make before we go, is that one of the good things about this is that the nitrates and the phosphates and the potassium, none of these things are ending up leaching and going down the river. So there's like a nutrient neutrality aspect to this. So if you're in a sensitive area, if you're in a built-up area, if you've got a concern from the ecologists about impacts of rivers from planting, these are the tools by which we can start to change these topics of conversation. So nitrate vulnerable zones are because they've already got enough nitrates in the waters. But nitrates in the water for two reasons. One is it's synthetic and it leaches, and the two is there's not enough fungi to hold on to the nutrition. So by putting in fungi and putting in the nutrition that is more akin and natural to working with the fungi, it then doesn't end up leaching and going down the river. So for those that have those sensitive planting projects, catchment sensitive zones, urban, ecologist engagement, these are the conversations that we need to start having, is the use of fungi and natural sources of nutrition. So I'm quite conscious of time. Really thank you for joining me. Thank you for Green Tech for hosting and for being our distributor. And yeah, you can have your day back. Happy Friday, everybody.
Lovely. Thanks a lot, Daniel. And yeah, I'm sure we'll catch up shortly.
Thank you.